accepted a lunch invitation. So I won't be able to stay for your talk, but I wanted to sort of log on at the beginning and at least be able to introduce you.
Yeah, thank you.
How are you doing?
I'm doing well. Yeah. Things are going pretty well. Family's healthy and yeah.
Fantastic.
Yeah, we've been out here in Pennsylvania since I graduated almost. So
Yes, yeah.
it's
And
been almost
you've
5 years.
been, it sounds like you've been very successful there.
Yeah, I've been fortunate to work in a really good lab, and had the opportunity last year um, to apply for and accept a position as assistant professor. So something I never really thought I would do, but here I am. So
Yeah.
yeah.
Well, that's great. So I guess you're probably busy doing things like writing grants.
Yep, exactly. I already submitted one in June of this year. It's supposed to have been scored yesterday, study section.
Oh,
So
which
um,
study section did it go to?
uh, they changed the names, and I'm not very familiar anyways, but it was CDMA, I think was the one.
It's like an
So
imaging.
it was a cardiac focus, but not necessarily imaging. I mean, we do have some imaging in it. So yeah. But and then I have a renewal that I'm working on for the current grant that I inherited, that we're hoping to submit in March.
Mm-hmm.
So yeah, things are going fairly well, I would say.
Fantastic.
Yeah. How about you? Still doing well in the lab, I presume?
Yeah, I'm very busy these days because I'm director of the Institute of Engineering and Medicine.
Mm-hmm.
After Dr. Chen retired.
Mm-hmm.
And also I'm also busier because Jeff also retired.
Oh, did he?
Yeah, he still comes in and helps out a bit with Cardiac Atlas, but he's it's only about once a week.
Okay.
And the other thing that's happened is we started a whole new research area on elite human performance.
Yeah, I talked to Jen about that not long ago. So that sounds really interesting.
Yeah. So the main, for us, I think the main difference is we work on a lot on skeletal muscle now instead of cardiac, but we still use all the same tools.
Uh-huh. Okay, so you're still using continuity for that.
Yeah, although actually probably continuity is one of the ones that's a little bit less commonly used. We're doing quite a lot of agent-based modeling and network modeling, and the musculoskeletal field has some pretty good software for finite elements and like bio and stuff. So
yeah,
some of my postdocs prefer to use that.
So are you working with Jeff Weiss at University of Utah for FE Bio? And
so we're not actually directly collaborating with him, but I keep mentioning the fact that we know him well, and
yeah,
that anyone who needs help with FE Bio should be able to get it.
Very cool.
Yeah.
Oh, that's awesome. Yeah, it sounded like a really cool project. I'd love to hear more about at some point.
Yeah.
So
yeah, well, we need to bring you back here for a visit so you can give a department seminar. And
that'd be great. I miss San Diego a lot, especially when the weather starts turning cold like it is right now.
Well, it's beautiful here.
I bet. Always is. And your kids are probably, let's see, your youngest was I think 12 when I left, so he's probably a senior in high school by now.
No, he's actually a junior in mechanical engineering and here at UCSD.
Oh wow, okay, so yeah, miscalculated.
And Jeremy's a PhD student at Stanford. He's actually doing biomechanics.
Wow, oh, that's awesome. So does he use FE Bio too?
No, although he might have ended up working with some of that. His PI does work with the Human Performance Alliance.
Does he?
But he's actually mostly working on constitutive modeling of brain.
Oh, okay. All right. Very cool.
So Kelly, we can't see anyone here, so we don't know whether we're talking to ourselves or to a whole room full of people.
Can you hear me, Dr. McCullough?
We can hear you, but we can't see anything.
Okay. We can hear you and see you just fine. I don't know how to make you see the room.
If you go to
Would be nice for Dr. Carruth to be able to see the room.
We're consulting our technology experts,
Probably just turn the
which is
camera
not
down.
me.
You see the arrow by the stop video?
Mm.
Oh.
By the stop video.
Yeah, turn the background off.
Yeah. Then should be, yeah, I think it's the background, but
Yeah, give the virtual balance.
yeah.
That one?
I can just turn it off.
Turn it off.
There we go.
Thank you,
Okay,
Daniel.
that's much better. So we actually have a whole room full of people here, Eric.
Yes.
Okay.
great.
So it's my pleasure to introduce today's sort of alumni speaker. Dr. Carruth was a PhD student in interfaces program, and he worked on cardiac structure function relations, and particularly using cardiac diffusion tensor MRI to characterize features of cardiac muscle structure and architecture that had previously not been identified by cardiac MRI. Now, actually, there's a lot of people interested in using diffusion tensor MRI for a lot of the things that he started doing in cardiac muscle and other tissues. But it was very much a multi-scale project because it involved sort of histology, and it involved tissue scale imaging, and it involved microstructural modeling and data science. After he graduated from our program in bioengineering, he moved to Pennsylvania, where he's at the Geisinger Health Research Institute, and he's recently become an assistant professor in the Department of Genomic Health. And so he's going to tell us about how he uses imaging and genomics and the multi-scale approaches he learned in the interfaces program to study cardiovascular diseases. I apologize that I can't stay for his whole talk, but I'll hand over to him now so that I don't use up too much of his time. Eric.
All right. Thank you so much, Dr. McCulloch, and thank you for the introduction. Thanks for the invitation to come and talk. This is really exciting for me and somewhat nostalgic. I remember sitting in that room eating pizza or other delicious lunch and listening to other students talk about their research. So it's been 5 years since I left. Feels like yesterday. So let me share my screen here. Hopefully I get the right one. And can you let me know if you can see my screen?
We can.
Okay, excellent. So I kind of want this to be fairly informal, so feel free to interrupt me with questions. And I'll also try to make sure there are occasional breaks where you can ask questions. But yeah, feel free to interrupt me. So just a quick overview, a very high level. I'm going to talk mostly about the research that I've done on a disease called arrhythmogenic cardiomyopathy from a genome screening population. And then towards the end of the talk, I'll also talk a little bit about how the interfaces program at UCSD helped me at the time I was a graduate student and into my early career to now, as well as what I think all graduate students should know. So this is something that maybe a lot of you already do know, but we'll talk about it anyway. So before I go into the research, I have to acknowledge the many individuals who have contributed to this work over the past 5 years plus. And to you, these are just nameless faces, but each of these individuals has been critical to this work, so I can't talk about it without acknowledging them, as well as the funding mechanism that has made this work possible. So as I said, I'm going to talk about a disease called arrhythmogenic right ventricular cardiomyopathy, or ARVC. I used a different acronym earlier, and I'll talk about why that is in a few slides. But this is a progressive disease that's characterized by fibrofatty degradation of the muscle tissue in the heart. And this can manifest in right dominant or biventricular forms. And when I say the right, I mean the right ventricle. For those of you who are less familiar with cardiac anatomy, there's a left ventricle and a right ventricle. ARVC, it typically affects the right ventricle, but can also affect both. And on the right here, you can see this histology slide showing the fibrotic remodeling in blue that replaced the muscle tissue, which would appear in red. And you would typically see, if you took a section out of the heart, almost all red, which is healthy tissue. So this is a rare condition. Its prevalence estimates range from 1 in 1,000 to 1 in 5,000. And it's heritable. So over time, as people have come in with this disease, genetic testing has revealed a desmosome gene variant in over half of cases. And the genes that are primarily involved include these 4: PKP2, DSP, DSG2, and DSC2. I won't go into detail about what each of them does, but they're all different components of a structural component of cardiac tissue called the desmosome that links adjacent cells together. So ARVC is one of the leading causes of sudden death in the young, particularly in athletes. And the mean age of onset or initial clinical exam for index cases is around 36 years of age. So what are the clinical outcomes of this condition? At the extreme, we have, like I just mentioned, sudden cardiac death, which has been reported as a presenting symptom in about 1/4 of index cases. And up to 20% or 1/5 of all sudden cardiac death in individuals less than 35 years of age has been attributed to ARVC. So this is an important condition to understand to hopefully prevent sudden cardiac death in these young, healthy individuals. But importantly, if we are able to identify individuals who have this disease early enough, then they actually have a somewhat favorable prognosis if we include interventions such as implanting a cardioverter defibrillator, which will detect an abnormal electrical rhythm in the heart and shock the heart back into a regular rhythm, as well as lifestyle modification, including restriction from vigorous physical activity, which has been shown to make this disease progress faster, as well as prescription drugs for antiarrhythmics. And so I do have to clarify a quick point on terminology. You've already heard me use a couple of different terms on this slide, and it really has many different names that refer to different aspects of the disease. Historically, ARVD was the first recognized clinical entity and was described as a dysplasia because it was thought to be a congenital condition. Over time, it was recognized that it was more of a cardiomyopathy, so most people made the switch in the literature to ARVC. Some people who really clung on to that dysplasia use a combined term of ARVD/C. And then more recently, over the past 15 to 20 years, it's really started to be recognized that this isn't just a right ventricular condition, but that it can also be left dominant, or called arrhythmogenic left ventricular cardiomyopathy. And as recently as 2019, the Heart Rhythm Society published this consensus statement defining arrhythmogenic cardiomyopathy generally as an arrhythmogenic heart muscle disorder not explained by ischemic, hypertensive, or valvular heart disease. And so this really encompasses both ARVC and ALVC and biventricular disease. Again, I'm going to focus to begin with on ARVC, but we'll talk about ALVC and ACM throughout the talk. So I just want you to understand how those are related. So why is Geisinger a good place to study this? So Geisinger is an integrated health system that has a service area shown here on the right that covers central and northeastern Pennsylvania. It has a number of different hospitals, urgent care clinics, etc. But despite it covering mostly rural areas, Geisinger really has advanced technology. Interestingly, Geisinger was one of the earliest adopters, health system adopters of the EPIC EHR, electronic health records system. So we have longstanding electronic health records available here with a longstanding enterprise data warehouse since 2007, a group that's closely connected to the research department here called the Phenomic Analytics and Clinical Data Core that assists with data governance and modeling. And we also have a very stable patient population with an outmigration rate per year of less than 1%. So the patients who live in this area stay in this area, which means that we have longitudinal data on these patients over a long period of time. Additionally, and these are just a couple of pictures. The bottom one here is the building I'm sitting in right now. Additionally, in 2007, there were some researchers here that started a really visionary project called the MyCode Community Health Initiative. This is a research study that had the ambitious goal to sequence over 500,000 Geisinger patients. To date, over 320,000 patients have signed the consent form to participate in this study. And through a collaboration with Regeneron Genetics Center called Discover, 184,000 individuals have had exome sequencing completed. And those exome sequence data are tied to the electronic health records that I mentioned on the previous slide. So this is a really valuable resource for research purposes. We can investigate genetic variants in the exome sequencing data and link that with health record data. On top of the MyCode Community Health Initiative, there's also this, what's called the Genomic Screening and Counseling Program. So rather than just having this data for research purposes, we have the ability to identify variants, pathogenic or likely pathogenic variants in genes that are associated with certain conditions and notify the patients that they have this genetic variant after they're confirmed in a clinical laboratory. And then those patients can follow up with healthcare providers to hopefully mitigate the risk of actual disease. Some of the conditions include breast and ovarian cancers, some of the common ones that are high risk, but some of those are also inherited cardiomyopathies such as ARVC. And you'll notice the 4 genes that I mentioned earlier are included on this list. I'll also point out the inclusion of this FLNC gene, which I'll talk about a little bit later on. So I've kind of been rushing through that, but any questions at this point about Geisinger or ARVC? Okay. When we started this project, really the vision was to use these resources to pioneer what's called a genome-first approach to arrhythmogenic cardiomyopathy, or ARVC. So the traditional sort of clinical pathway in ARVC was you'd have, a young, seemingly healthy, maybe active individual who may have vague symptoms, and then suddenly they'd have a cardiac event, either resuscitated from sudden cardiac arrest or experience a sudden cardiac death. And if they survive that event, then they would see a cardiologist who would then prescribe treatment or lifestyle intervention, etc., as I mentioned before. But using genetics, we hope that we can identify individuals who carry pathogenic or likely pathogenic variants in genes that are associated with ACM and notify them. They can then have imaging done, electrophysiology studies, and if they express the disease, then can route them to a cardiologist to hopefully prevent any sudden cardiac event from occurring in the first place. But in order to do this, at the time, there were several questions that we really didn't know. We knew how common these variants were in individuals with disease and their family members through the traditional clinical pathway, but we didn't really know how common these variants are in a general population. So that's really the first question that we sought to answer with this project. And from there, these individuals could really fall into two main buckets. The first is they could be what's called phenotype positive. That is, they satisfy TFC or task force diagnostic criteria for ARVC. And we didn't know, how common that was. The term for that is penetrance. How common do people who have a genotype actually express the phenotype? And what are the factors that mediate that penetrance? On the other hand, these individuals could be phenotype negative. They could not express any disease. And in this case, there are also questions such as, is this complete non-penetrance? They'll never go on to express disease. Have we just caught them too early before they actually develop any symptoms or any phenotype recognizable through standard clinical workup? Or is it a novel phenotype that we haven't really considered? And how can we distinguish among those different cases? So those are the questions that we sought to answer using these data that are available to us. And to do that, we really took a two-pronged approach that both stemmed from the MyCode Community Health Initiative. So the first, as I mentioned, is using this long-standing electronic health records database to investigate genotype-phenotype associations computationally. And we looked at these pathogenic or likely pathogenic variants in desmosome genes. And we also have looked at other cardiomyopathy-associated genes, including Titin and, again, this FLNC. And then the second approach is, or second part of this approach is following the return of these genetic results to patients through the genomic screening and counseling program. They'll have these clinical evaluations that we can further assess, after they are actually receive a workup for ARVC, do they express disease? We can also recontact these patients, and we've been doing that and performing exercise history surveys to understand if this is a factor in the development of disease in this context. And finally, Andrew mentioned that I did a lot of work in advanced cardiac MRI as a PhD student. And so we can invite these patients in if they don't express a phenotype and see if we can detect subclinical features of disease using advanced imaging. Did I hear someone wanting to say a question or a comment? Okay, I'm not hearing anything. All right, so the first two questions that I'll talk about that we tried to address are first, the prevalence of these variants in a general population and the likelihood of expressing disease in those patients. So we published a couple of papers several years ago now about this, and we learned that the prevalence of these desmosome gene variants it's actually much higher than we would have expected, at around 1 in 435, I think it was. And if we can compare that to the disease estimates that I talked about earlier of 1 in 1,000 to 1 in 5,000, which is quite rare. And the flip side of that is, we use this Bayesian inference equation to estimate the penetrance of disease, and we estimated it at about 6%, which was quite a bit lower than what you would observe from family studies of index cases who come in with disease. We also observed limited evidence generally of related disease in the health records. We found that about 15% of these people that have a genetic variant satisfied at least a minor task force criterion, but that was actually in proportion with controls. So when we did a phenome-wide association study in these individuals, we did not see any signal that rose above the line of significance here. We might have expected to see, you know, some of these red triangles in the circulatory system bump up above that line, but we did not. So the summary from those two papers is that the prevalence of desmosome variants in a general population is higher than we would expect based on disease-based cohorts. And the flip side of that is that the penetrance of desmosomal ARVC in general population is low at around 6%. But again, that's from electronic health records prior to these individuals being notified about their genetic result. So my slide's not advancing. Oh, here we go. So so after these individuals had a chance to follow up on their result clinically, we looked through their health records again to try to understand this better. Did they just maybe not have any information about their heart? And that's why perhaps the penetrance was low. So from the first 65,000 MyCode participants who were eligible for this genomic screening counseling program, 92 carried a pathogenic or likely pathogenic variant in one of those 4 genes I mentioned earlier, and they're color-coded by the gene, as you can see in the key here. Of those, 16 ended up indicating either that they did not want to follow up, or we could just never got a hold of them, and they were lost to follow up. So we had 76 who indicated that they would choose to follow up with a provider, but not all of those actually did. 14 we hadn't seen any documentation in their chart, and 3 indicated they may have seen an external provider outside of Geisinger. So we ended up with a cohort of 59 who had an observed evaluation in their health record. And of those 59, most of them, 38, had no diagnostic task force criteria for ARVC, whereas 21 at least had a minor criterion. Again, as I mentioned before, those weren't too uncommon and were consistent with controls. But there were some individuals who expressed more than just a single minor criterion and actually were categorized into a couple of different buckets here. So on the right is this ARVC that we classified in the beginning as the right-sided or right-dominant disease that has the official diagnostic task force criteria associated with it. And we found 4 individuals who were classified as either definite or borderline ARVC from this effort. Interestingly, though, there were 6 individuals who we classified as having left dominant disease. That is, they had left ventricular dysfunction and arrhythmia concomitant with that. And one of the interesting things here is there was some overlap in these 2 individuals, really had biventricular disease. And you'll notice that 6 out of the 8 that are in these 2 buckets were carriers of DSP variants. And we also characterized some of the imaging findings from these patients. 3 out of these 4 are DSP carriers. This one in the top right panel is a PKP2 carrier. But you'll notice that they all express this similar pattern on late gadolinium enhancement imaging of having fibrotic remodeling in the midwall of the left ventricle in sort of this ring-like pattern, which has been observed in other studies of especially DSP carriers. So from this, we confirmed the prior finding that penetrance in a general population of desmosomal ARVC is low. Our rates were consistent with the 6% estimate from our EHR study. And we also observed that ALVC is at least as likely as ARVC in this context, especially for DSP and possibly DSG2 carriers. All right, so one of the things that we're starting to investigate more recently, we had evidence about this in one of those early papers, is whether the prevalence of genetic variants differs by population. And so in one of the early studies, we saw that individuals who are genotype positive in this cohort were less likely to be of European ancestry compared to the rest of the MyCode cohort here in central Pennsylvania. And so we've expanded that analysis with more individuals in the MyCode study that have gotten sequencing data, as well as comparing across a few different biobanks. So we have MyCode here on the left. We have the All of Us study, which is highly focused on diversity within the United States. We have the Penn Medicine Biobank, which is a biobank in a health system that's really a referral center for more advanced disease. And we have the UK Biobank, which is also a general population based biobank, again, in mostly European individuals. And as you can see, the individuals who are carriers of desmosome variants, and especially PKP2 variants, are much more likely to be of African ancestry in MyCode, All of Us, and in the UK Biobank. Whereas in this Penn Medicine cohort, we don't see that effect, and we think that that's due to the ascertainment differences in that cohort. So this is a short but important result that we've observed, and we're working on publishing this at the moment. So from that, we've concluded the prevalence of PKP2 variants is higher in African ancestry individuals. We have not yet observed any difference in the phenotypic burden by ancestry, but that'll remain to be seen as more data is investigated. All right, so I want to pause again. I haven't heard any questions so far this whole time, but maybe you guys are done eating and can ask some questions.
Questions? Not yet? Oh, we got one.
Okay.
Yeah, for the imaging, what specific imaging are you using? Are they CT scans?
So these were cardiac MRI that were done clinically. And so late gadolinium enhancement is a method to inject a contrast agent and then wait for about 10 to 15 minutes for it to diffuse into the cardiac tissue. If you see these bright spots are indicative of fibrotic remodeling, and LGE is typically used to investigate coronary artery disease that's associated with heart attacks. But you'll typically see that, like here, this purple area arrow is pointing to an area that's probably from an ischemic region, whereas in non-ischemic disease, you'll see these arrows pointing to regions that are closer to the epicardial surface of the tissue, the outside edge of the ventricles. So yeah, these are cardiac MRI.
Okay. Is it radiologists doing the diagnosis, or do you have a quantitative way to do it for the just?
Yeah, so for these were done in the hospital settings. These were read by a radiologist. For this research paper, however, we had a one of the cardiologists is an expert in the inherited cardiomyopathies, and we had him review these to make sure that the radiology report was accurate, and some of them he did correct. So good question. Any others? All right, hearing none, I'll continue on. All right, so around the time that we were coming up with this project and really getting it off the ground, people started recognizing that individuals with arrhythmogenic cardiomyopathy were more and more often expressing, or it was more and more frequent to identify mutations in this gene called FLNC. I may say filament C because that's the protein that it encodes. So this was one of the seminal papers that talked about this association from disease-based cohorts. And this is a figure from that paper showing in the top left corner, you can see this fibrotic remodeling on a post-mortem photograph of this individual's heart, corresponding with the late gadolinium enhancement that we just talked about on their cardiac MRI before they died. And you can see this, again, this ring-like pattern of late gadolinium enhancement in the left ventricle. Here, this pattern extends from the base of the heart all the way down through the apex in different regions, and histology shows again this blue infiltration of fibrotic remodeling amidst the purple or red muscle tissue. And this individual died suddenly at 17 years of age after a soccer game. So again, we had a unique opportunity to study this gene and its association with arrhythmogenic cardiomyopathy in a genome first manner, which, you know, is a really unique strength of the MyCode data. So we did just that, and by the time we did this study, the number of MyCode participants with sequencing data had really expanded up to 172,000 individuals. And from those 172,000, 60 were identified as carriers of loss of function variants in this gene FLNC. And when we looked through their electronic health records to identify whether they had left ventricular dysfunction and or arrhythmia, we found that they were much more likely to have dysfunction, as well as dysfunction and arrhythmia, which is the definition that we used to define left dominant arrhythmogenic cardiomyopathy, could also be called ALBC here. And again, this was from the health records, which perhaps are incomplete. And so what we did in this case was we had those same cardiologists I mentioned a minute ago review these charts of these 60 individuals, as well as 120 matched controls from the genotype negative MyCode population. So these were age and sex matched to the filament C group. So in this diagram, the white boxes represent a yes to the question on the left, the gray represents a no. So you can see that 56, almost all of the 60 carriers had some cardiac evaluation available in their electronic health records. It was also a majority in the controls, but not quite as high a percentage. And of those 56, 10 actually had ventricular dysfunction, which was, I can't remember the full definition, but included things like a reduced ejection fraction on imaging. And that was significantly higher than 4 out of the 103 individuals in the control group. And so if we break down, if we look further at those 10 individuals about their ventricular dysfunction, that could have been caused by any number of different diseases. So we looked to see if they could be explained, whether or not they could be explained by other non-cardiomyopathy comorbidities. And if the answer here is yes, then that means this is a non-ischemic, you know, non-hypertensive condition, not cardiomyopathy. And then if they're highlighted in orange, then they also had arrhythmia as part of the presentation of dysfunction, which would be a good definition of arrhythmogenic cardiomyopathy. So we observed 3 out of the 10 who actually did have arrhythmia as well. These 2 did not have arrhythmia, but that's not to say that it couldn't have been picked up if more testing had been done. And it's interesting to think about whether the filament C variant could have been a contributor in these 4 individuals who had dysfunction and arrhythmia, but they also had some other comorbidities that may have confounded the diagnosis here, which was quite different than the larger group of 120 matched controls, who all of them had some other comorbidity to explain that away. If we also looked at the available echocardiogram data in the electronic health record of these individuals, we found that the left ventricular ejection fraction was significantly lower in the filament C loss of function variant carriers compared to the non-carrier controls. And so from this, we confirmed that filament C loss of function variants are associated with ACM, and primarily left-sided disease, with penetrance that was somewhat higher actually than the desmosomal ARVC that we observed in prior studies. So this was a cool finding, and the paper that we published had a really nice editorial comment from one of the worldwide experts on filament C. So that was a cool experience. All right, so back to this sort of diagram outlining the questions that we're seeking to answer here. The next question that we've been trying to answer over the past several years is, we observed that there was low penetrance for desmosomal ARVC in this genome first cohort, but it wasn't zero, right? There were some individuals who did express disease. And so we wanted to understand what are the factors that distinguish those individuals from others. And as I mentioned a couple of times, oh, careful with the scroll wheel. I'm going the wrong way. Okay. As I mentioned a couple times before, history of physical activity, especially participation in high intensity or high endurance athletics, has been associated with ARVC diagnosis. So this was one of the papers that really showed that by one of our collaborators, Cindy James at Johns Hopkins. And she showed with her team that if you break down the population of index cases and their family members by whether or not, by how much exercise they've done in their lifetime, that the more exercise they did, the more likely they were to meet diagnostic criteria. And participation in endurance athletics was also, you're also much more likely to meet diagnostic criteria than if you did not. And so we copied their approach by using the same phone survey that they did. We worked closely with their team to do this work, and we surveyed the individuals in the genome first ARVC desmosomal gene variant cohort at MyCode, or at Geisinger through MyCode. And we observed in the Johns Hopkins cohort, which is the disease-based cohort, updated since this time, 2013, but we observed that same difference, that if they were in the TFC group, they had much more prior history of physical activity, whereas in the disease negative cohort, they didn't have as much. In MyCode, however, we didn't observe that difference. And so there are several things that could explain this. One of which is, like I said, the penetrance was low, so we didn't have a large number of cases who actually met task force criteria. So our statistical power wasn't great for this study. Also, the distribution of variants across the desmosomal genes is quite different in a disease-based cohort versus our genome first cohort. So in the disease-based cohort, it's like 70% PKP2 and much less common in the other 3 desmosomal genes. Whereas in our cohort, it's much more balanced across those 4 genes. So those are 2 of the reasons that may be contributing to this, but we think that this is encouraging for providers who are seeing patients after, secondary findings or incidental findings of genetic variants, that they can say, well, you're most likely okay to do this high intensity or endurance exercise if you want, but more data are needed to really solidify that. All right, now the final question that we're going to talk about is, what about these individuals who are phenotype negative? And can we use advanced cardiac MRI to really understand whether this is non-penetrance, pre-emergence, etc.? So a prior study has looked at this in, again, disease-based cohorts, individuals with diagnosed ARVC, and they've shown that this parameter called native T1 is quite different in patients with overt disease compared to controls, and even they're seeing a trend for this native T1 to be higher in at-risk relatives. These are relatives with genetic variants. And so what is this native T1 time? So basically, the tissues in your body all have different response to being magnetized, having a magnetization, and the relaxation from magnetization is what this T1 is. It's a relaxation time constant for your tissues, and it really is a measure of the difference in tissue properties. So higher native T1 is associated with diffuse fibrotic remodeling, which, as we know in ARVC, is one of the characterizations of that disease. So it stands to reason that native T1 would be higher in these patients and their at-risk relatives. But again, we don't really know what that looks like in a genome first cohort, at least until we did this study. So the way we did this was a little bit challenging. We had to look back in the health records here and identify from our genotype positive, phenotype negative cases, which patients had a cardiac MRI that included native T1 imaging, which isn't always done. These patients had to have no other cardiomyopathy associated gene variants and no phenotype, no cardiomyopathy, no ischemic disease or sarcoidosis, which is another sort of related disease. And the really challenging part was identifying the controls. And so what we did for that is we looked through the health records again to identify cardiac MRI studies that were physician interpreted as normal. These individuals also had to have no known cardiomyopathy genetic variants. They weren't necessarily in my code, so we can't say for certain that they did not have one, but it's quite unlikely. And they also could have, they could not have any cardiomyopathy, ischemic disease, or sarcoidosis. And the reason for the CMR study could not be to rule out ARVC or ACM. And so we looked at the following parameters here on the right-hand side from the cardiac MRI to see if we could observe any differences. The main ones that we'll focus on are, again, this native T1 and atypical late gadolinium enhancement that we've talked about already. So the late gadolinium, stumbling over my words, late gadolinium enhancement was much more common in the genotype positive group, even though these individuals weren't classified as having any ACM, they did have atypical late gadolinium enhancement on their MRI compared to none of the control group. And that was a significant difference there. And again, you would expect that if there's fibrotic remodeling in these patients, the native T1 time would be increased, and that was indeed the case in these 18 patients compared to controls. So we think that there's a real signal here, and we're working on expanding this analysis. Since the time we did this study, more patients have gone through the clinical workup. So we think that expanding the number of patients will really solidify these results. And so the other project that we're working on with this is to use what's called strain imaging. Some of you may have heard of different types of imaging to investigate deformation, displacements in cardiac tissue or other tissues. One of the main ones is called cardiac tagging, and this one, DENSE, is displacement encoding with stimulated echoes. It's a very similar technique where we get a magnitude image and 3 phase images that correspond to the X, Y, and Z coordinates in the scanner. And so these phases are directly encoding the displacement of the tissue. So a bright region here is that this point has moved significantly inward. A dark region is that this point has moved significantly upward, also inward. We can code those displacements in 3D space. This is a 2D mapping, but you can imagine the third dimension going through the screen here. And then from that, we can map those onto a grid and compute what are called strains. So this is a measure of the deformation of the tissue. And it's been shown in prior studies that strain is a much more sensitive measure of cardiac function compared to what's typically done, which is ejection fraction, just measuring the volume of the chamber at different time points and how effective it is at pumping the blood. So again, I've showed this before, but this is how we track those points through time. This is the magnitude image, and the points are overlaid on that image to show you how they track with the tissue. All right, and again, these are preliminary results here, but in the first 7 individuals for whom we have the analysis completed, we've observed that the strain values from this imaging seem to be reduced compared to the strains that we've observed in healthy individuals using the same or very similar techniques. And so, like I said, we're continuing to work on this and haven't run any statistics on this yet. But I'm optimistic based on what we're seeing so far that we'll actually have a signal here to report. So from those 2 studies, we have evidence that there is subtle subclinical abnormalities in these genotype positive, phenotype negative individuals on advanced cardiac MRI. So we think that this is a potential method for really fine-tuning our ability to discern patients who are truly at risk for a sudden cardiac event. As I said, the next steps include continuing those imaging studies by expanding the native T1 or myocardial mapping analysis from clinical scans. We're also expanding our research-only strain imaging protocol. We're currently scanning patients to finish that up and running analysis simultaneously. And let's see, I'm also working on a grant renewal to expand this work in the future by using deep learning to assist providers in diagnosing these individuals by really running models on the ECG and echocardiogram to identify patterns that are machine recognizable, but maybe not human recognizable. And we also want to understand, we saw that exercise maybe didn't play a strong role in the disease development in these patients, but we think that there may be a polygenic component, meaning that you may have the combination of many common variants maybe predisposes individuals to getting disease on top of the single rare variant. All right, so that finishes up my portion about the research that I've done. So again, any questions about what I've shown so far?
I have a question about the deep learning. So, what sort of programs do you use for what deep learning?
I had a hard time hearing that.
I'm asking about the deep learning. Can you hear me now?
Yeah.
Yeah, so could you comment a little more on that? What programs do you use to do that? And then also maybe what are a couple of the features that you think they would be looking at in the echocardiogram?
Yeah, I can't speak to it a lot because we haven't done it yet. This is very early stages of planning for writing this grant proposal. So there are several models out there, many models actually, to really dive into ECG data, especially, and echo data. The common tools that we use are in Python using like scikit-learn, PyTorch, and those kinds of things. But there's also, Google's TensorFlow. So those are all the common programs and tools that are used. The models that are commonly used are convolutional neural networks, but we're working with our collaborators at Johns Hopkins who have shown that these there's a new type of model called variational autoencoders that we think are showing some promise. And what they have shown is that rather than being a complete black box, so to speak, you can start to use the output of the model to go back and sort of reverse engineer an ECG signal based on the results of the model to show like, okay, we think that this is classifying this ECG in this bucket because of these features, which is really valuable and hasn't been really present in prior machine learning models. So again, I can't answer in a lot of detail about that yet because we haven't done it. But yeah, it's something we're hoping to dive into.
Thank you. I was just wondering if you could say a comment on what features you think you could feed back into that model.
Right. Yeah, exactly. So I don't have any slides for this, unfortunately, but some of the so ARVC in particular, there's ECG is one of the hallmark tests that are done to assess diagnostic criteria. And some of the things that are included in that are things like what are called epsilon waves. So if you look that up, if you Google epsilon waves, it'll tell you that's the most specific finding on ECG that's associated with ARVC. However, the challenge with epsilon waves is they're very, very challenging to identify. And different providers will say, yes, that's an epsilon wave. Others will say, no, it's not. And so there's a lot of disagreement about it. So we think that a machine could potentially tease out those signals a little better than even experts in cardiology. So that's one example, but there are probably many that we don't even know about that we think that a machine can probably identify that we can't.
Thank you.
Dr. Carruth, I have a question. I was wondering if you could comment on whether there's any commonly used preclinical models of ACM, whether they reflect the same genotype and part form.
Okay, I had a hard time hearing that too. It sounded like you're faded out a bit at the end, but you're asking about commonly used clinical models of ACM?
Yeah, I was wondering if there's any commonly used preclinical models, whether they reflect the same genotype and part variants in patients.
Oh, preclinical models. That's a good question. I don't know of any. And like I said, this is kind of this genome first approach has really been pioneered by the work that we've been doing. So, it's been a question that has been on a lot of people's mind for a long time, but we just haven't had the data to really investigate it. As you can see, it's a rare condition, so having a large enough cohort of genome screened patients has been lacking until recently. So yeah, we think that this is potentially promising. We did, identify patients who didn't know beforehand that they had ACM, and several of those in one of those early papers actually did get an ICD placed, which is a quite invasive procedure, and has a lot of what's the word I'm looking for, challenges associated with it for the person that receives it. But yeah, so I don't know how to answer that question, to be honest. But we think that this is probably one way to start to identify these people earlier.
Thank you.
Anybody else? Do you mind answering a few questions about how the program has helped your research and or career?
Yeah, so I can move on to that. So
there we go.
got a few minutes left here. Interfaces was a really great program for me. I used the same term for this. To interface with students across different disciplines was really valuable, and really, for me at least, enabled expanded opportunities. I was able to work with neuroscientists doing the diffusion tensor MRI that Andrew mentioned. I worked with cardiologists for doing imaging of mouse myocardial infarction. And a lot of that was facilitated by the interfaces program. So I was able to really connect and learn how to communicate across different disciplines. And I've learned that interdisciplinary research is really where true innovation occurs, when you're able to associate with and communicate with individuals from other departments, other disciplines, you really get a lot more insight into the type of work you're doing. Another benefit was you get frequent practice presenting research, asking and answering questions at seminars like this one, at the symposium. That is actually really beneficial. And I know as a graduate student, I tended to kind of shy away from those opportunities because it felt overwhelming at times. But you know, having that practice really helped me hone my presentation skills. I don't think I'm an expert still, but I think I've come a long way from that practice. So don't shy away from those opportunities, even if you feel like, well, I don't have much to say. It's helpful to present your ideas and get used to it. Obviously, having the T32 training grant available, that helped support my research. I don't know if that's still a thing, but if it is, look into it and try to take advantage of it. Having the specialization for multi-scale biology on my diploma, I don't know that it helped in terms of, you know, my career and getting a job, but for me, it was a source of, you know, pride that I did this extra thing, and I don't know, it felt good to have that on there. And then some maybe less career focused benefits were this seminar series was really nice to sort of break up the week, think about other topics in science, and again, just talk to other people, get out of the lab and do something different. They say there's no such thing as free lunch, but the Interfaces program has proven that is not true. So that's always nice too. And as a graduate student, you know, having that is just a small little thing that goes a long way to making your week a little better. And then, so what I've listed on this slide is what I think all graduate students should know. So for me, you know, I, as a graduate student, actually struggled at times to feel like I was doing, you know, really good work and to feel like I was progressing. I was a first-gen college grad, which may have contributed to that. I think it's common in the sciences to be introverted in nature. And that, on top of the fear of embarrassment or failure, led to me not being great at communicating with my advisors, and perhaps suboptimal performance. I think if I were to go back, I could have done better by just being more open about what I didn't know and being willing to kind of just be open about that so that I could learn. You're really there to learn, and so don't be afraid to, again, ask questions, be a little bit vulnerable, and you'll learn a lot from that. So one thing that I've started doing in my career and that I would recommend to anyone who's not doing it currently is just set up weekly meetings with your advisor. If you don't have that already, it's vital to help you get feedback on your work and help you with vision about where you should go and how you should do those things. I started doing that later than I should have in my PhD, and the difference it made was astronomical. Again, science is best interdisciplinary, so learn to work with others, communicate across disciplines, like I talked about on the last slide. And the final point is that, you know, life is not all about work. I'm a Christian, so I've put this reference to Luke chapter 2 at the bottom. This scripture and the essence of it has really helped me shape the balance of my life. I focus on these four main areas: intellectual, that I include learning new things, my career, and the finances for me and my family. Physical includes eating well, getting exercise, getting outside. You've got that amazing La Jolla Shores beach, go for a surf every once in a while. Spiritual is really important: prayer or meditation, whatever that looks like for you. And social, make sure you spend time with family and friends, because even though, you know, graduate school is this really intense period of study, you want to get done as quickly as you can. You'll never get those years back, so make sure you focus on identifying your priorities make sure they're balanced across those. And yeah, I hope that helps a little bit with some of you to find a little more meaning and joy in your graduate career. So finally, I do want to say thank you for your attention. If interested in this work, I am hiring currently, particularly for individuals who are well-versed in imaging. If you're interested, please reach out. I've left my email here and my LinkedIn profile where you can reach out to me. So yeah, and then even though we're at time, any final questions?
Thank you so much, Dr. Carruth. We really, really, really appreciate it. I appreciate especially the ending part, you being so honest about struggling and offering some advice to the students and trainees for that. And I encourage everybody to join our LinkedIn page, which is on the website. And let me know if you have any questions, especially if you have any suggestions to, for what to eat for lunch. And who else you'd like to see speak. And thank you again, Dr. Carruth, so much for your time. Right.
Yeah, thank you all for listening and being a good audience. Appreciate it.
Thank you. Anybody got anything before we stop recording? Okay. Thanks again, Dr. Carruth. Enjoy the rest of your day.
Yeah, you as well. Thank you.
Bye-bye.